In this year's title, you're being asked to design and carry out an investigation related to membranes, osmosis, and food preservation. Now, the live brief that was released a few weeks ago states that foods with high water content support microbial growth, and that preservation can involve removing water from microbial cells by osmosis. And your job now is to investigate one or more aspects of this topic. And a supporting line from the brief would be investigate one or more aspects of this topic. Your research question really does need to be very specific. It should be narrow, measurable, and testable. A good question focuses on one factor affecting osmosis or food preservation. And examples that you might use here would be the effect of either temperature or solute concentration, or maybe even surface area. The way to come up with an idea for your research question would be to start off by identifying the variables that affect osmosis that are mentioned in the brief provided. So, starting point here would be temperature, concentration gradient, and surface area. Then you need to choose one of these that you can safely and realistically test in your school laboratory. Some of the areas of research that you could begin to look at would be the effect of salt concentration on osmosis in plant cells, so maybe potatoes or onion cells, the effect of sugar concentration on food preservation, how temperature affects the rate of osmosis. You could look at how surface area, for example, the size of potato cubes, affects osmosis. You could look at the difference between using salt and sugar as preservatives. And one more idea or example could be to look at investigating water loss in fruit or vegetables in different brines. Now, brine is a supersaturated salt solution, so you might vary the concentration of salt here. In short, yes, two students can investigate the same factor, but the caveat here would be that each student has to produce their own independent work as required by the authentication rules, and your teacher should be able to go through this with you in more detail. If you look at the brief, it says that the work ultimately submitted to the SEC has to be the candidate's own individual authentic work. If you want to do well in the introduction section, I would include a clear title, a short explanation of osmosis and food preservation, your research question or hypothesis, and why the question matters. And what you need to do here then is link it back into the live brief and bring in an example of real-world relevance, i.e. food preservation using osmosis. The research section should include the following: scientific theory on osmosis and membranes, how solute concentration affects water movement, how osmosis is actually already used in food preservation. So you could bring in tuna in brine, you could bring in fruit in sugary syrups. You should also include secondary data from reliable sources, and this brings in your background research. You have to have a piece on the evaluation of your sources. So how reliable are your sources? Why did you trust that particular source? And you need to learn how to carry out proper referencing. And again, if we take a line from the brief here, it says that the background research needs to include some secondary data, some evaluation of that secondary data, and also you need to include referencing. When it comes to research, the first starting point should be your textbook. The textbook is often overlooked as the first point of call, but that's where I would encourage my students to go back and revise the topic of osmosis and movement of water through membranes. So your textbook would be the starting point. From there, you should move on to reputable online sources. And this might include university websites, scientific organizations, government food safety agencies. So good sources of information or good sources for your background research in your biology and practice investigation would include, again, I would recommend you all to use your school textbook first. Then you should be looking up some peer-reviewed articles. I then might look at Teagasc, the agricultural website. I might look up the Food Safety Authority of Ireland, the World Health Organization, reputable university websites, and some scientific encyclopedias. Now, I would add in here that I would avoid the following. I would avoid using blogs. I would avoid Wikipedia at all costs. And I would avoid looking at random websites or AI generated content unless it is properly referenced. Now, just on that, you are allowed to use AI for some of your background research, but it must be referenced. So if you use it and then you don't reference it, this could essentially be a form of plagiarism, and it could cost you marks. So in relation to recording your findings, I would encourage you all to use an investigative log. Now, that's a fancy term for what I'm getting my students to use, which is just a small copy, a 120-page copy that you might have even had in primary school. Write the phrase biology in practice on it, and that should be where you jot down your ideas, you write down your thinking at that moment in time, you write down every single thought that you have, your references, and it should be your little Bible that you go back to when you start typing it up. So in the SEC brief, they say your investigative log can be used to keep a record of your thoughts and your research references. So this would avoid you writing random things down on scraps of paper and then losing them. So if you get yourself a little copy, put a heading on it, investigative log, biology in practice, and keep that with you at all times when you're doing your work on the BIP. So when it comes to designing and planning your experiment, the first thing that you need to know is what your variables are. So what's your independent variable? What are you changing? What is the measurement that is going to depend on your independent variable? And obviously that's your dependent variable. And then you need to be aware of all the other variables, and you need to fix them or control them. So if you were to investigate, for example, the effect of surface area on osmosis, you might want to keep temperature as a fixed variable. And then you might say, okay, I fixed the temperature using a water bath, and it'd be really, really important here to get even more precise than that, and to say something like, I fixed the temperature at 25 degrees using a thermostatically controlled water bath. After that, you would need to write down the list of equipment or apparatus that you require. It is then up to you to write down your step-by-step method. The way I like to think about this is it's kind of like a recipe for making a cake. You need to write down every single step in a logical sequence so that if you gave this method to somebody else, they could easily carry out your own experiment. You need to be aware if there are any safety considerations. And if you were to go down the route here of seeing maybe how much microbial growth occurs in a food item based on osmosis, you need to be aware of the aseptic or sterile technique, and you need to exercise care when handling microorganisms. You would also need to be very aware of fairness, keeping conditions constant and trying to remove any bias. And the last point here would be you'd need to link it back into stage 2 of the process, which is your background research. How did your research influence your design would be a really important consideration. So the definition of a hypothesis is that it is an educated guess based on observation or research. To put it another way, a hypothesis would be a testable prediction based on scientific theory. And so an example that might link directly in with our BIP this year is increasing salt concentration will increase the rate of water loss by osmosis. And then you find a way to test that. So in relation to the theory or the research to include in your hypothesis, this is again where you go back to the background research that you would have done, especially the part explaining how water moves from low solute concentration to high solute concentration across a semi-permeable membrane. You show research influence design by explicitly linking the research to your decisions. So for example, you should be saying things like, my research showed that temperature affects osmosis. So I kept temperature constant at 25 degrees Celsius using a thermostatically controlled water bath because this is the optimal temperature for bacterial and fungal enzymes to work at. So the difference between a control variable and a constant condition would look something like this. A control variable is something, a factor that you deliberately keep the same, like temperature, using the water bath that we outlined. A constant condition might be a background condition that just naturally stays the same. So for example, I use the same type of beaker or I use the same type of conical flask. The areas that I recommend you focus on when conducting your experiment would be accuracy, correct and safe use of equipment, safe working in the lab, fair testing where you are trying to avoid bias, and one of the most important pieces will be the recording of clear primary data. Now remember, primary data is the whole point of the BIP. You need to collect data yourself in the lab, and that's what primary data is. When it comes to improving the accuracy of your results, the following are things you need to take into consideration. Use precise measuring equipment. So don't use a beaker when a graduated cylinder might be more accurate. Carry out repeat trials, also called replicates. So if you get a set of results, repeat it a second and a third time, and then take averages, and this will increase your percentage accuracy. Try to control your variables tightly. If you're saying the temperature is at 25 degrees, but it's really at 21 degrees, that's going to have a big impact. Use consistent sample sizes. Use the same number in your samples. So a big sample might be 10 or 12. Try not to use just 2 or 3. And if you can, try to avoid human error. Read scales and meniscus at eye level so you avoid the error of parallax. When it comes to repeat trials or replicates, you need, I feel, to have at least 3 repeats. This is the standard for reliability, but as usual when it comes to these experiments, more is better if time allows. Your method should be really, really detailed. Another student or a random person in the school should be able to repeat your experiment exactly by reading your method and following the instructions. There should be no ambiguity, and the way that you write it should be really clear and really concise, and you should absolutely use bullet points. Justifying equipment means that when you choose a piece of equipment, you give a reason for it. So an example here might be, a digital balance was used because it provides more accurate mass readings. Or you might say, I used a digital balance that was accurate 2 decimal places because it's more accurate than a balance that is only accurate to 1 decimal place. It's really important to conduct your experiments safely and to be aware of your own safety and the safety of other students around you. You should absolutely make yourself familiar with the lab rules and follow them. At all times, you should be wearing safety goggles and a lab coat. You need to handle your salt and sugar solutions safely and clean up any spills immediately. You need to dispose of food samples properly. And if you do go down the road of counting microbial growth, you need to be really, really aware that working with microorganisms can be hazardous, and you need to employ something called the aseptic technique and incinerate or burn the microorganisms at the very end. So in your biology and practice investigation, you can use images, and the SEC tells us that a student can use up to 20 images total. Now, I would say that is not necessarily a target. I would encourage you to use images where appropriate. I wouldn't just go firing in 20 images just to reach that number of 20. If you put in pictures of your lab setup, if you put in pictures of graphs that you maybe have drawn or screenshots of graphs that you have generated, maybe using Microsoft Excel, that would be a very appropriate use of images. I don't think you necessarily need to have the full 20, but I definitely would include some. So in the data and analysis section, it's really, really important to have a lot of information here. So the first thing would be your primary data, and this should be included in what we might call raw data tables. So it's essentially what you wrote down on the day that you did the experiment. The next thing that should come after that would be the processed data. So this is you taking your primary data and maybe carrying out a mathematical process on it, which could just simply be getting the average of your repeats or your replicates and looking at things like percentage change. From there, you should take that data and generate a graph. And be really aware then of what your dependent and independent variables are when you're labeling the axes on your graph. And then in the analysis section, you're going to look at your graph and your tables, and you're looking for trends and patterns. And then this is where you need to come into your own and put on your thinking cap, because then this is where you have to carry out interpretation of your results. So The live brief that the SEC provided said you need to include experimental observations, data presentation, and also data analysis. So in relation to graphs and calculations, I feel that you should be including things like line graphs, which could show the rate of osmosis over time, or bar charts, which might be more appropriate for showing mass change. You should show any and all calculations that you do. So this could be percentage change calculations, and you should also include your calculations for averages and ranges. When evaluating your data, you need to first of all be looking for patterns. And then you need to link your data back to your question or your hypothesis, and you need to see and ask the question, does my data support my hypothesis? Then you need to look at the reliability of your own results. You need to highlight any anomalies. So if the hypothesis is not supported and you get an erroneous result, you need to actually record that. And then you need to actually look as well for possible sources of error, and you need to own them. There's no such thing as bad data. If you get data that you feel isn't right, and remember there's no such thing as right or wrong data in science, you need to actually record that. If you have unexpected results, you need to remember that there's no such thing as bad data, and you need to try and explain them honestly by suggesting reasons. There was some experimental error, there were some equipment issues, there was biological variation, and then you need to discuss how you could improve the experiment if you were to do it again. So there's a huge piece here in the BIP about reflection. So you need to look back on what you did and ask the question, how could I improve it if I was to do it again? It's really important to link your results back to your research question. You need to try and directly answer the question with your own data. So an example here might be, my results show that increasing salt concentration increases water loss. Therefore, my research question is supported. So when it comes to the reflection piece at the end, and if you're looking to say how your experiment could be done better, I would focus on maybe the following. If I was to do this again, I could use more precise equipment. I could increase the number of repeats, which might increase my percentage accuracy. I could test a wider range of concentrations or temperature or surface area. I could try to improve my temperature control. And then maybe you could say something like, I could use more consistent sample sizes. So in relation to errors or improvements a student could list, some of the things that would come to mind for me might be the use of inconsistent sample sizes. You could say I used a ruler or a cork borer. There could be temperature fluctuations. Instead of using a thermostatically controlled water bath, the student might have just heated up some water and left it in a beaker. And then as time goes by, the water temperature would begin to drop, and therefore the temperature would not be kept constant. There could be measurement errors. So you could be using a regular thermometer, and then you might say, I would use a digital thermometer if I was to do this again. Sometimes if your containers are left open, you will get evaporation of water, and this will affect your results. So I might suggest here covering the containers with lids. And then another one that could come up might be timing inconsistencies. You need to be very, very careful here and use a stopwatch, maybe set an alarm on an iPad or a Chromebook, and just make sure that if you're saying that you left it for 30 minutes, that you left it for exactly 30 minutes and not 29 and not 31. When it comes to the conclusion of your BIP, it should include a direct answer to the research question that you provided in your hypothesis. You should then go on to say whether the hypothesis was supported or not. And then you should go into a very short summary of your key findings and then link it back to real world relevance. Link it back to the brief which talks about food preservation or the use of food preservation in the real world. So in relation to osmosis being used in real life examples to preserve food, I would be suggesting that you could use the following. Salting fish or meat, so tuna in brine would be a good example here. Sugaring fruit for jams or marmalades. Pickling vegetables, so pickled gherkins, pickled onions. Using brine to preserve cheese. or even dehydration in food storage. So if you think of things like packet soups, it's completely dehydrated and then you just add hot water and it rehydrates the food. So it's really important to reference properly, and there are two main types of referencing that are used nowadays. You've got the Harvard referencing method and the APA referencing method. They're not going to be overly strict on the use of those two methods, but what I would say that you need to include would be the author or the organization, the year that it was published, the title, and then the name of the website or the book. If you're accessing content online, you need to include the entire address or URL, and it's good practice to include the date that you access the material. So the brief requires referencing of all sources, including AI. And the brief clearly says candidates must reference and acknowledge their research sources, including any use of AI. So to avoid plagiarism, you absolutely must reference any material that you access, including your textbook. So my final tips for completing the Biology in Practice report would be, first of all, to relax and realize that you have almost a year to complete this. This video has been recorded in early March 26, and you don't have to submit this until February 26th, 2027. Try to embrace the scientific process here and be curious and take time to think and to plan. You should spend about 2 of the 6 weeks allocated for this to your initial response to the brief and the background research. So don't feel under any pressure to have the experiment done during 5th year. Okay, in case my own class, my class will be doing their experiment in October, and we'll be doing the planning between now and then. In relation to the report itself, stick to the 1,500 word limit and do not go over it. In order to do that, that means you need to keep your writing really clear and really concise. And what I say to my students here is that bullet points are your friend. You do not need to write big, long paragraphs. Use images only when they add value. You do not need to reach the upper limit of 20 images. Keep your log updated. So remember, I mentioned your investigative log. Get yourself that 120-page copy. Any ideas, any thoughts you have, write them down. If you access a website, if you read a book, if you read a paper, write down the name, the author, the year, the date that you actually read it, and then that can go into your references section at the end of the brief. In relation to references, remember, they do not count as part of your 1,500 words. And I will be saying that if you want to do very well in this, you need to show a lot of research, therefore you need to show a lot of references. Now, what's a lot? I will be suggesting 8 to 10 references. Okay, that will be my own personal opinion on how many references to use. Proofread your Word document before you turn it into a PDF and before you submit it to the SEC. You will have to sign off on it, and your teacher will have to sign off on it as well. It's really important then that everything is your own work. Follow the section headings the SEC have provided, and the very best of luck with the work.